Thanks for your moderator and Magnus Group also for providing this opportunity. My title, Abundance of Aero-algal Allergens and Public Health Hazards in Pandu River Ecosystem. First of all, I would like to introduce you from our workplace, Pandu River. Pandu River is one of the river of Gangetic alluvium, and there is no record of its algal profile and biological significance. A lot of the research work has been done on river Ganges ecosystem, but the river Pandu is known as it is overshadowed because of river Ganges in Kanpur, India, famous for pristine purity of water and sentimental and religious forever beliefs. Present observation record aero-algal species of Pandu River basin and portray its importance in biology of algal ecosystem and spread allergens. Algae are ubiquitous and omnipresent organisms growing profusely in varied environments. Airborne algae remain viable in the atmosphere for a long time after reaching a suitable environment where conditions for these for their growth are well start germination. Allergenic algae are dispersed effectively by air in dry condition in form of algal spore or fragments. Dispersed algal allergens cause either contact, uh, skin or respiratory allergies among human beings and cattle. The airborne algal flora and its potential allergenicity is a primacy for intensive investigation. There are few investigations scattered in the literature while aeroalgae quantitative data value of algal growth and infestation in Pandu River, Pedbin, Bingwa, Kanpur, and Shorapur, Fatehpur is lacking. Present study enriches the available literature and fill this vacuum in order to obtain and evaluate pertinent information relating allergenic form of algae and their prevalence in the air. Present observation record aero-algal species of Pandu River ecosystem and portrays its importance in spread of allergenic diseases. The marked resemblance between allergenic algae in air around Pandu River and its water record cyclic transmission of aquatic and aero-algal allergens in air and riverine systems. Present investigation emphasizes implication of aero-algae causing prospective public health hazards and its control. Aeroalgae is an emerging science and has wide ramifications in aerobiology and pollution. Aeroalgal allergens have been reported from North America by Louis 1977, and excellent work by Brown and others 1964 presented information in some depth on the abundance of heterogeneity of airborne algae. In India, some experimental studies on airborne algae were made from Aurangabad, Tilak and Rishi, 1978, Delhi, Mittal and others, 1979, 74 and 73. Nagpur, Marathi and Reddy, 1980 and Bijapur, Mane, 1980. Aeroalgal allergens also have speaking language. Aero-algal allergens also have been reported from Indo-Gangetic Plain, Shukla 1983 and Tripathi, Gupta and Tiwari 1990. Algal allergens of Kanpur, UP include 39 species belonging to 9 genera. Present observation record aquatic algal species of Pandu River basin and emphasizes their implication in public health hazards in combination with aero-allergen hither to not recorded. Aero-algal species were collected by gravity slide sample method. The sample were kept at 12 selected sites in Pandu River basin between Panki and Bengwa. Glacial jelly was used as adhesive in microslides. Up to 24 hours slides were examined under the microscope for identification of aeroalgae collected. The process was repeated after 2 weeks by placing new slides on various selected sites during the various calendar months. Apart from this monitoring, aeroalgae was conducted using exposure of petri dishes of inch diameter containing 10 ml Bennett and Chow 10 medium for 24 hours, once in 15 days throughout the year at the same time. Petri dishes were then kept in lab in BOD incubator for 30 degrees centigrade and exposed to fluorescent light intensity 375 lux for 18 hours per day for 1 month. The growth of the algae into media was recorded after taking out alga sample, scales, and identified. The allergenicity of algae was probably convincingly first demonstrated by C.S.J.M in Wisconsin. He reported hay fever, conjunctivitis, and asthma in patients sensitive to blue-green algae. McCullough first reported an inhaled allergy due to airborne blue-green algae. There are also reports of algae causing hay fever elsewhere. C.S.J.M. Incidence of allergenic disease due to algal allergens belonging to chlorophyceae and cyanophyceae have posed serious public health problems. Air carried rich in algal flora and contained algal allergens during the month of August to November. Cyanophycean from outweigh chlorophycean allergens during present investigation. This might be due to the relative greater resistance of cyanophycean algae to unfavorable conditions. Quantitative evidence of allergenic form is directly linked with spread of allergenic diseases. Allergenic problems caused due to such algae are rhinitis, asthma, hypersensitivity, pneumonitis, bronchial propagation, mortality in fish, cattle, and animals, coupled with symptoms of partial paralysis, loss of balance, general weakness, and photosensitization of skin. Allergenic algae in river Pandu between Panki and Lingua include Chlorococcum humicola, Chlorella vulgaris, Sandesmus arquatus, Sandesmus abundans, Sandesmus acuminatus, Sandesmus bijuga, and Sandesmus dimorphus, Sandesmus obliquus, Sandesmus cordicorda, Anamina cercinalis, Anabina spherica, Anabina planktonica, Anabina fertilisma, Anabina oscillatoris, Nostoc lincia, Nostoc muscorum, Nodularia, Spumigiana, Oscillatoria anguina, Oscillatoria limnatica, Oscillatoria princeps, Oscillatoria subveris, Oscillatoria animalis, Oscillatoria formosa, Microcystis aeruginosa, Palmidium fragile. Allergenic significance of Kanpur algae has been emphasized earlier by Shukla 1981. Algal allergens exhibit a specific periodicity in area. There are 24 species belonging to 17 genera of aero-algae recorded between Panki and Vingwa, out of which 14 algal species are known as causal only for spread of allergenic diseases. Some of them are only found in certain periods of a season or seasons, while others may grow throughout the year. Periodicity and quantitative abundance of such Algae determines the extent of potential allergenic diseases spread, and knowledge of such may be help to devise control measure. Results indicate that 24 algal species were recorded as aero-algae comprising Chlorococcum humicola, Chlorella vulgaris, Sandesmus abundans, Bezuga, Sandesmus obliquus, Spirogyra species, Culenca radiata, Pediastinum tetrans, Osis solitaria, Cyclotella, glomerata, Gumphonemia species, and Anabina spherica, Anabina oscilloides, Aphanothesis species, Gliotracta natans, Microcystis aerogenesa, Nostocomni, Nostoclincia nodularia, Oscillatoria anguina, Oscillatoria princeps, Oscillatoria formosa, Oscillatoria subraris, Formidium tenuo from Sinophytes. Out of these, allergenic algae which exhibit the growth in area and are also significant propagator of allergenic disease comprising Chlorococcum humicola, Chlorella vulgaris, Sandesmus abundans, Sandesmus bezuga, Sandesmus obliquus from Chlorophyceae, and Anabina oscillatoides, Anabina spherica, Microcystis aeruginosa, Nostoclincia, Nodularia, Oscillatoria, Princeps and Anguina, Oscillatoria formosa, and Oscillatoria subraris from Steinophyceae. The observation suggests that algae comprising in Pandu River basin are transmigrated through the agency of wind and the river water recede. Some of the algal fragments keep blowing with the wind. On return of a suitable environment, survive again. Such aero-algal transmigration is substantiated through observation of algae in exposed patches containing Banach medium, Banach and Chow medium. Allergenic algae which cause problem in human beings such as asthma, rhinitis, bronchial propagation, pneumonitis, hypersensitivity, conjunctivitis, and hay fever. In case of the animal partial paralysis, mortality, photosensitization of the skin, general weakness, and loss of balance. The 14 algae which were found during the experiment are here with their camera lucida, which are Chlorella vulgaris, Chlorococcum humicola, Sandesmus, and Sandesmus bejuga, Sandesmus obliquus, Nostoclincia, Anabina oscillatoris, Anabina spherica, Oscillatoria princeps, Oscillatoria subraris, and Oscillatoria formosa, Oscillatoria anguina, Nodularia, and Microcystis from Steinophyceae. Airborne algal flora and its potential allergenicity is a promising area for intensive and interdisciplinary applied research. Present observations pinpoint the need to undertake exhaustive studies to record aeroalgae at different elevations. The results are of pivotal significance in formulation of an aeroalgal allergy calendar for assisting clinical and therapeutic treatment of patients of bronchial, skin, and eye infection. Thank you. Thank you.